the very beginning stage of cellular respiration is what we refer to as glycolysis. And glycolysis is a little bit unique in that this is the one part of cellular respiration that's going to take place out here in what would be the cytoplasm of the cell. So in this eukaryotic cell, as I've drawn it here, this is my mitochondria. And for the first stage, the mitochondria is not going to be involved at all. And again, that's unique because we tend to usually think of the mitochondria as being the organelle that's responsible for cellular respiration. And it definitely will be for the second and third stage, just not for the first one. So let's talk a little bit about glycolysis and what's going to happen with glycolysis. As far as the overall result of glycolysis, we're going to start out here in the cytoplasm with an organic molecule. In this example, we're going to take glucose. And that glucose is going to get broken down into two molecules that we call pyruvate. And this very term here, glyco means sugar and lysis means splitting. So we are splitting the sugar quite literally because glucose happens to be a 6-carbon molecule and each of these pyruvates happens to be 3 carbons. So we are splitting it apart. If we just talked about kind of the overall things that are going to happen during this process, we are going to begin to oxidize the food. And by food, I'm talking about the organic molecule. As we go through it, this is specifically going to be glucose. So we're going to begin to oxidize it. And we're also going to make just a little bit of ATP. And that is important because ATP is really the end product that we're looking for in all of this, because with cellular respiration, we do want to really release the energy from food so that we can utilize that to make that valuable short-term energy storage molecule. So as we begin that process and we look at how we're going to reach that more unstable state, the first thing that's going to happen is the cell is going to spend an ATP molecule that it already has. Now, when it does that, it is going to basically take the 6-carbon molecule. So I'm going to write my 6 carbons down here again. And what we're going to do is we're going to take a phosphate off of the ATP that we're spending, and we're going to attach it right to the end of this glucose molecule. So when we do that, ATP goes in, and out of the reaction comes ADP. So that right there would be spending some energy or investing some energy in this overall process. And now what we have done is we have generated right here a molecule that is more unstable. And if it is more unstable, remember that stability is related to the potential energy. So it has more potential energy when we're looking at it there. The next thing that we're going to do is really a very similar step. We're going to do the same thing. We're going to take that 6-carbon glucose, which now has a phosphate on one side that we just put on there. And we are going to, again, spend an ATP. And when we spend the ATP, this time we're going to put the phosphate on the other side. And so ATP goes in, and ADP comes out because we have removed a phosphate group from our ATP. And now what we have is a very unstable molecule. And so this time, that molecule is highly unstable. And if it's highly unstable, it has even more potential energy. And what that means is that that molecule wants to split apart from each other. So that is what's going to happen in this very next step. So just to put some names along with this molecule that we're looking at right here is what we call fructose 1,6-bisphosphate. So again, this is a highly unstable molecule, and it's going to split quite easily at this point just because it is so unstable. So in the very next step, that is exactly what we're going to see happen, is we're going to end up with 2 3-carbon molecules, and they do have different names to them, but each one of those 3 carbon molecules is going to have a phosphate attached to it. Okay, and these 2 molecules, one of them is going to be what we call pGal, and the other one is going to be what we would call G3P. So if we write those names on here, I'm going to call one of them pGal, and that's phosphoglyceraldehyde. And this one is going to be what we would call G3P, which is glyceraldehyde 3-phosphate. They're very similar molecules to each other, but those are the initial splitting products when we split our glucose molecule in half. At that point, we have reached the end of what we call the energy investment phase of glycolysis. And as the energy investment phase, at this point in time, we have spent really, so we have spent, or you could say invested, if you will, 2 ATPs. We have actually utilized 2 ATPs that we already had in the cell so that we could actually get this sugar, this glucose molecule, to split. So we utilized one here and we utilized another one here. And if you look here, we have not actually yielded or produced any ATPs yet. So at this point, we would be kind of negative 2 ATPs if we were actually keeping track of our yields. If we take a look at what we're going to do now with those 2 molecules that we have, these 2 split molecules, we do have an enzyme that is going to be able to really flip these 2 forms back and forth with each other. And anytime we have an enzyme that can generate isomers, that is going to be called an isomerase enzyme. Okay, so we do have an isomerase enzyme that is going to be able to flip the pGal into a G3P, and it can flip G3P into pGal. What it's actually going to do is it's going to take both of those and it's going to turn them into 2 G3Ps. Okay, so we will ultimately, when we finish this first stage, we're going to have 2 G3Ps. Okay, so I'm going to write those right here. And now let's take a look at what's going to happen to those. Again, those are my G3Ps. Here I'm looking at just one of our G3P molecules. Each one of these G3P molecules is going to go through the same steps, so I'm just drawing one of them for simplicity purposes. The first thing that's going to happen to this G3P molecule is it's going to go through a redox reaction. And since this is a redox reaction, remember that one of the overall things that glycolysis is going to do is it's going to oxidize this organic molecule. So as we begin to oxidize this organic molecule, we are going to see an electron carrier, and our electron carrier is going to be NAD+, and NAD+ is going to come in, is going to grab some electrons, and when it does, it turns into its reduced state, which is NADH. Now recall that NADH, since it has the electrons, it is going to have more potential energy than the NAD+ would have. So that is a valuable molecule because it is holding on to those valuable electrons. When we do that, we're going to have our 3 carbon molecule, which has lost some of the potential energy because it has lost some of its valuable electrons. It still has its phosphate group attached, and it's actually going to get another phosphate group. We're going to have just this extra phosphate group that's hanging around in the cytoplasm that comes and gets attached here as well. So now we have this molecule here, which is partially oxidized because we did collect some electrons. And then what we're going to have is we're going to have an ATP synthesis occur. And this would be what we would call a substrate level phosphorylation. So what that means is that ADP is going to come in, and it's going to end up getting one of these phosphate groups that's attached to this organic molecule. So when ADP gains that phosphate group, it turns into ATP. What we're left with after that is going to be our 3 carbon molecule that still happens to have a phosphate group attached to it. 1 phosphate group attached to it. We do have a name for any enzyme that performs this kind of transfer of the phosphate group. These tend to be what we call kinase enzymes. So kinases are involved in what we would call phosphorylation. And that's what this one happens to be doing. The next step is going to involve another similar reaction. And so we're going to take that, and we are again going to take a phosphate group, and we're going to transfer that phosphate group onto ADP. So ADP comes in, we transfer a phosphate group to that, and it leaves as ATP. So now our phosphate group is no longer here on this 3-carbon molecule, and that 3-carbon molecule is going to be pyruvate. Now, some of the steps have certainly been left off of this, so I don't want to leave the impression that I have drawn every single step of this process. We usually tend to divide glycolysis into 10 individual reactions, so I know I haven't shown all of those. But what I have shown is kind of the key steps where we see either oxidation reactions or we see ATP being utilized or being made. So if we look here and we talk about what happened, in this picture, you're seeing that we made one NADH. Okay, so we reduced that molecule. And we also see a plus 2 ATPs because we actually made those. I will remind you that this was for 1 G3P, but remember that we had 2 of these up here because that was per glucose that we started with. So if we wanted to talk about kind of where we're at energy-wise, we would have to take these numbers here and multiply them by 2. So we're going to have 2 NADHs and 4 ATPs that we just made. And if we talk about the overall profit from glycolysis, remember that earlier we spent 2 ATPs. So that was kind of a negative 2 ATPs. So if we subtract that off, overall, what we end up with is a profit, or what you could call a net, of 2 NADHs. NADH is again holding on to those valuable electrons. It does have extra potential energy because it is holding on to those electrons. And we have 2 ATPs profit. Okay, and this would all be sitting out in the cytoplasm because that is where the process of glycolysis takes place.